Folks, welcome to my channel. In this video, I'll be discussing about the types, functions, the enzymes present, and the clinical significance of lysosomes. So there are three forms of lysosome, and these are the primary lysosome, the secondary lysosome, and the tertiary lysosome. So here we have a diagrammatic representation of the formation of the primary, secondary and the tertiary lysosome. So the primary lysosome or the Golgi hydrolase vesicle is a vesicle which buds off from the Golgi complex, that is the primary or the first formed lysosome. This lysosome may join with an endosome bearing special membrane proteins which enter the cell through the process of endocytosis, and there will be fusion between this primary vesicle and the endosome to form the secondary lysosome or the endolysosome. Whereas if the primary lysosome is combining with the phagosome, which is formed by the foreign matter being phagocytosed into the cell, then we get the phagolysosome, which is also known as the tertiary lysosome. The functions of lysosomes are many, and these include the digestive. It acts as a digestive organ of the cell. So lysosomes contain many enzymes which are essential for the intracellular digestion. The second function is a bactericidal activity. So lysosomes kill and remove infective organisms and foreign bodies, and lysosomes are highly bactericidal. And the three mechanisms are the two enzymes which are present, that is the lysozymes and lysoferrin, as well as the next function is autolysis. So when a cell is damaged or ruptured, the enzymes liberated within the cytoplasm digest the cytoplasmic contents, a process known as autolysis. The autolytic enzymes are mainly lysosomal enzymes. The next function is the recycling of cell organelles, where the worn-out cell organelles are transported to the lysosome by the autophagosomes and are then transferred into the interior of lysosomes, where there will be action of the lysosomal enzymes, and that undergoes a process called autophagy, leading to their recycling after degradation. Here the nutrients liberated from the digested organelles are reused by the cells. Fertilization of the ovum, it is due to the acrosome, which is a lysosome, that is a modified form of a lysosome, and then the next and the final function would be the removal of products of metabolism. Now let's see the lysosomal enzymes. So the lysosomal enzymes, they have an optimum pH around 5, as already mentioned, and these enzymes are mainly polysaccharide hydrolysing enzymes, and the examples will be alpha glucosidase, alpha fucosidase, beta galactosidase, alpha mannose, beta glucuronidase, hyaluronidase, aryl sulfatase, and lysozyme. And the next one or the next group of enzymes present are the protein hydrolysing enzymes, and these include the cathepsins, collagenase, elastase, and peptidase. You should always remember that cathepsins act as a marker enzyme for lysozyme or the lysosome. Now nucleic acid hydrolysing enzymes such as ribonuclease and deoxyribonuclease, and the lipid hydrolysing enzyme such as fatty acyl esterase and phospholipases. Now the clinical application of lysosomes. In gout, the urate crystals are deposited around the knee joints. So these crystals, when phagocytosed, cause physical damage to lysosomes and release of enzymes. Thus, inflammation and arthritis result. So this is mainly due to the extrusion of the hydrolytic enzymes, which are acidic enzymes that are present within the lysosome. The similar reason applies for the postmortem autolysis, where following cell death, the lysosomes rupture, releasing the hydrolytic enzymes which bring about the postmortem autolysis. Now let's talk about the inclusion cell disease or the eye cell disease. It is a rare condition in which lysosomes lack in enzymes, but they are seen in the blood. So this is a protein targeting defect. Here the enzymes are synthesized but are not able to reach the correct site. It is shown that mannose-6-phosphate is a marker to target the nascent enzymes to lysosome. So in these persons, the carbohydrate units are not added to the enzyme, and mannose-6-phosphate deficient enzymes cannot reach their destination, thus it forms a protein targeting defect. So here, the enzymes which are supposed to be incorporated with the mannose-6-phosphate is not incorporated, and thus there is deficiency of these carbohydrate units in the nascent enzymes. And the mannose-6-phosphate is a marker which helps in targeting the enzymes to the lysosome. As a result, due to its deficiency, the enzymes do not reach the lysosome, and the lysosomes are devoid of the enzyme, whereas blood has greater concentration or the enzymes which are supposed to be present within the lysosomes. Now, lysosomal proteases, cathepsins, are implicated in tumor metastasis. As I've already mentioned before, they act as the marker enzymes for lysosome. There are a few genetic diseases which are associated with the deficiency or absence of the lysosomal enzyme. Silicosis, which result from inhalation of silica particles into the lungs, which are taken up by the phagocytes. So lysosomal membrane ruptures. So this is also the extrusion of the hydrolytic or the acidic enzymes which are present within the lysosome to the cell or towards the cytosol of the cell, and this stimulates fibroblasts to proliferate and deposit collagen fibers, resulting in fibrosis and decreased lung elasticity due to cell death. That's it for the video. If you honestly did understand the topic, do like, share and subscribe. Thank you.